Hello, everyone, and welcome to today's keynote presentation, Artificial Intelligence for Medical Imaging with a live Q&A. This webinar is part of the Cancer Research and Immuno-Oncology Virtual Week. And I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the left of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, just click on that help desk button located at the bottom of your screen where the navigation bar is located and from the lobby. I now present today's speaker, Dr. Hugo Aerts, Director of Artificial Intelligence in Medicine program and an associate professor at Harvard University. For a complete biography of our speakers, please visit the biography tab at the left of your screen. Dr. Aerts, welcome. You may now begin your presentation, sir.
Thank you. Thank you for the introduction. So in this talk, I want to give you a high-level overview of different AI approaches that we're evaluating in medical imaging. So the AIM program, the AI medicine program at Mass General Brigham and Harvard Medical School, is a program that's focused on evaluating AI technologies very close to the clinic, right? So where can we make a difference? We are, focused on various different fields. So we do oncology a lot, but also cardiovascular imaging. We even are working in diabetes and general health. But in this talk, I will give you some examples of other areas, but I will mainly focus on the cancer applications. So these are my disclosures. All right. So first of all, go into why AI in medicine, right? So what is important? Where can we see breakthroughs, and where do we expect very big barriers? Then I will give you some examples to give you a flavor of the studies that we're doing and the problems that we face. And at the end, I want to take a couple of minutes to really dive a little bit deeper into what we think how the field as a whole should be moving forward. So we'll give some recommendations there. All right. So first, I'm going to talk about why AI in medical imaging. And what is really interesting to me is that also this field, again, is following very nicely this Gartner hype cycle. So, about 10 years ago, there was this technology trigger, it was even longer for deep learning, right? And what happened afterwards was really amazing to see, right? These big conferences, everything was about AI and how AI will revolutionize everything. There was like big companies like IBM even saying that we should stop training radiologists. A lot of startups. I think it was the largest field of VC funding was in AI and medicine for many years straight. So I think now we're like a little bit over the peak and we're going down. And we see that first startups are starting to fail. We see that many results are not reproducible, so it's very difficult to actually show that these models are generalizable and can be actually applied outside of the training populations. And I think in general, people start to realize that it is very difficult to transform medicine, right? Medicine is a very, very difficult field, and it's very difficult to transform this. But overall, we'll reach a plateau of productivity at the end. What we can do today is something that we could not do, say, 15 years ago. We can very quickly develop solutions that have a much higher performance and are even better than humans in very specific tasks. And that is something that is real. So that there is a real revolution that is happening. But it's really interesting to see how this hype is actually, how we actually follow this hype cycle pretty nicely. All right. So what is how you can also see this is think about the AI applications along the cancer care pathway, right? So there's many different steps and touch points in this patient pathway where AI applications can intersect. And of course, you can think about examples where you have data, you have methods, and you have a clinical problem that then can be addressed. And there's only a couple of areas, and there's often these solutions only touch one or a limited number of these touch points. So think about, for lung cancer screening, so low-dose CTs are often acquired now, and the lung cancer screening programs are very active now in the US. There, of course, neural networks apply to these CT scans could really help with the identification of lung nodules and make that screening process more efficient and cheaper. And that is something where we see that certain methods from CNN networks together with the CT data is really driving certain solutions, and also Google is even working on in this field. But also in other parts, we can think about like the cancer genome data and using graph models, for example, that also will help there to improve the staging process. So what is really interesting there is that what kind of touch points will AI or is AI able to address, and also how high is the performance, and how can we make sure that these methods really make it into the clinic? And this is another way of showing this. So this is the oncological data stream, how we like to say it, where you can see that we're collecting more and more data over time. So the stream gets thicker over time. And there's a lot of data there that's available, but there's also a lot of clinical decisions that are being made for screening, for diagnosis staging, for treatment decisions, where we can use AI to really improve this whole process. The big, what also this is illustrating is actually an issue that we're facing a lot in the field, is that it's almost no patient, no two patients will have exactly the same data. So there's a lot of different paths through this, all of these different steps. And there's a lot of missing data. There's a lot of missing time points. So there's a lot of chaos, to say it like that, and it's very chaotic data. So that is a very big issue that we face, is that, if we expect that a certain application will help in a certain field, is there data? And then we spend a lot of time accumulating this data, and there's a lot of often a lot of missing data. And this is simply the clinic, right? Like, this is not something that we can change easily. Where our program focuses on is also, we are very early stage from, we think about new concepts and we test them in scientific studies and we publish about them. So very far from the clinic, but we also are all the way to running clinical trials with the technologies that were developed in the lab. And then you really run into completely different problems that we didn't envision before. So by doing this, we want to really bridge this translational gap, right? So, the cog from model development is turning well. There's many publications out there, and hundreds of publications are published every month in AI and medicine. So that wheel is turning very well. But there's not a lot that actually try to really focus on the validity, the usability, and the utility of these algorithms to really drive the clinical impact. And that is a very big gap to bridge, which we hope, and also that we see also other teams are now really working on that, so specifically focusing on these different aspects. All right, so this was like a higher level overview of what we are doing, and also how we see the AI in medicine. Now I want to give you a couple of example studies that we're working on to give you a flavor of the projects that we are investigating. So the first concept is about biological aging. So we know that people age in different rates, and we know that genetic and lifestyle factors really influence your aging process. So we have an hypothesis that your biological age is not the same thing as your chronological age, right? And this is, of course, very important in medicine, where there's a lot of clinical decisions being made on a person's chronological age. So if we can better quantify a biological age, then we can this will directly influence treatment decisions, especially in cancer. So think about it, if you have a 75-year-old that is actually as fit as a 65-year-old, then it is expected that this person will do better and live longer after cancer treatments than, say, a 65-year-old that is 10 years older. The biological age is 10 years older. So this whole concept of biological age gets now a lot of interest, and I think especially in medicine that this is something that we can, where we can really prove upon what we're currently doing. So what we said is that, what we wondered is your biological age also reflected in a simple selfie, right? So if you have a picture of an individual, right, is the biological age also reflected there? Then, and this is similar to, say, the eyeball test of a clinician's, or the, so it's a test, or like when a clinician walks into a room and sees the patient, they often really knows, okay, this individual is doing well or not. So our main hypothesis is that the biological age of an individual is actually reflected in a face photograph. And then we are doing research studies now to extract that information and also see what is the clinical utility of these. So we started to develop a deep learning algorithm, and here you can see Leonardo DiCaprio. That's a picture of him, we wanted to develop a deep learning algorithm that can accurately estimate a biological age from a face photograph, right? So that was our goal. So then, so we call this the face age algorithm. So face age. So then we had two different algorithms in series, where first we localize the face in the image, and then once we have this bounding box, we performed another very detailed quantification of the facial characteristics. And this is much more beyond what humans do, right? So the algorithm looks at a different way at aging than humans normally do. And the nice thing then is, at the end, you get a face age prediction. So in this example, Leonardo DiCaprio looks like 40 while he is 45. So he's 5 years younger than his age. So his biological age is 5 years younger than his age. So we used a large data set of 56,000 individuals that we made age balanced and manually curated. So we spent a lot of time to make it with ages similar to an older population that we would encounter in a hospital. And but we also curated to make sure that that the images were of high enough quality, and that also the face was nicely positioned in this. So then we had this algorithm we had it developed. Then we applied this to several clinical data sets. So one of the few professions in medicine that actually takes a standardized picture of a patient is radiation oncology. So in radiation oncology, you want to make sure that the right person is on the table, right? That you're, giving the radiation dose to the right person. So they always take a picture before treatment starts, and they check this every time that the patient is put on the table for the treatment. They make sure that it's the right person. So the nice thing is that these are often taken in a very standardized fashion, right? Stand against the wall, and they take, the same lighting. They take a picture using a similar camera. There's no filters, and there's no enhancements that are being made on these images. There's not few processing. And we have them before treatment starts. So we took advantage of data sets that we had access to. One was from Mastro, which is a radiation oncology center in the Netherlands, where we had almost 5,000 individuals with various stages and various cancer types. And then we had two different data sets from Harvard. So one focused on thoracic cancer, so lung cancer patients primarily, and also a palliative cohort, where we have very detailed clinical characteristics. And the nice thing is that these cohorts gave us opportunity to do a lot of different research studies. So we can compare your face age to your age. We can look at lifestyle factors. We can look at different stages and cancer types. But also we can investigate how well do these certain algorithms help, for example, the clinician in decision making. So first, focusing on the lifestyle factors, which I think is a very interesting result. So what you can see here is your face age minus age. So if the value is positive, then you look older, but if it's negative, you look younger, right? So then we went to a reference population, and these are considered healthy individuals that have no symptoms and considered healthy. What you can see there is that the vast majority of the individuals are within +5 to -5 years of their age, right? So very similar to their age, but there's outliers. Some of them are much younger, some of them are much older. But on average, it's very close. The mean is very close to 0. What is really interesting then is that if you look at the cancer patients, is that you already see the shift. So on average, we see that the cancer patients look 5 years older than a healthy reference population. And this is also expected, of course, because they are in a worse state. But it's pretty interesting that we can already derive this from the data. Then, if we look at smoking status, there's also something very fascinating here, is that we found that current smokers looked significantly older than never smokers, right? And this also makes sense, right? If you start smoking, overall your lifestyle of smokers is worse, and they are aging more rapidly. But what is very interesting there is that the current smokers also looked significantly older than the former smokers, but there was no difference between the former smokers and the current smokers, at least not a statistically significant difference. So this means that there's almost like a reversible effect, right? So when you start smoking, you start aging more rapidly, but if you stop smoking, there's also a reversible effect that can be measured in these photographs. Looking at BMI, we also did this. We didn't find a correlation. Actually, it was significant because it's so many data points, but you can overall see that there's a very weak correlation association between BMI and face age. So it's really capturing a different pattern in the data. All right. So then we look at prognostic performance. So we went to the master cohort of 5,000 individuals. And if you then put your face age and only look at face age, no other parameters, if you just put them in certain buckets, how old people look, you can really immediately see a very nice stratification, a very significant stratification of these individuals. So the older you look, the earlier you die. And we did also, of course, corrections for various clinical factors like age, gender, and tumor site. And this effect remains significant, right? So it is capturing, your face age is capturing something else that is not quantified in these parameters. And then we went to the Harvard cohorts, and we included for a very large number of parameters that we had available, like smoking, treatment information, tumor information, ethnicity, treatment attendant type, ER visits. All of this information was taken into account and corrected for, and the face age was still significantly predictive. So, the face age still added significantly to the model. So this really means that there's information in your face and how old you look that is actually not being captured in the current clinical parameters, and that is actually important for prognostication in the clinic. Of course, the nice thing is that now we have a very easy assessment of the health status of an individual, right? So we can put this into a app, and we can ask a patient to at home to take a selfie, for example, every day, or we do this in a standardized fashion when a patient comes to the hospital. But we have a very easy way to get information about the current performance status of a cancer patient at that moment. That is very cheap and minimally disruptive of the workflow. All right, so that was a bit about face age. So we can also, of course, extend this concept to other data types. And of course, in X-ray, right, there's a lot of information on an X-ray that tells you about the biological age of an individual as well. So what we wanted to do, and maybe also as a background, so the chest radiograph is the most commonly used imaging test in medicine, right? It's very, very frequent. And they often do this to see if there's basic radiological findings, right? Is there lung modules? Is there any even RIPS, right? Like they can assess these complications. But an X-ray is used for many, many different to rule out many different diagnoses and diseases. But what we wanted to see is that is there also a lot of information of an individual about their biological age, right? So for asymptomatic individuals that do not have any radiological findings, that got an X-ray, can we, for example, use a deep learning network to extract that information from these X-rays and then predict longevity, right? So, and can we also do this better than BMI and muscle mass and fat mass, for example? So that is something that we are now investigating. So first step in this was done by using two very large cohorts, the PLCO and the NLST, where we have about 60,000 X-rays of presumed healthy individuals that were included into these screening programs. So we used then a deep learning network to quantify the information on an X-ray and then predict how well will this individual, what is the prognosis of this individual. And what we could see is that and this remains significant after correction, we could see that we could very well identify a high-risk population. So a high-risk population of individuals that actually look much older on an X-ray than their peers, and this also remains significant after correcting for a large panel of clinical factors. And we also did a similar analysis in NLST, and we saw very similar results, right? So there is a subpopulation of individuals that do not do well that we can identify using these this biological age quantification of X-rays. But you might wonder, it's like, great, but what do you actually do with this? So we said, okay, but what now if we can put such a system in a hospital and use this to just automatically screen every X-ray that is that is being acquired, and then just say, okay, this individual is actually at risk for developing cancer, or this individual is at risk for developing cardiovascular disease, for example. So in the cancer example, and this is something that we published about 2 years ago, we actually said, okay, we want to now do this. We want to implement such a system. We want to predict who is at increased risk for developing lung cancer in the future, while there's no radiological symptoms at this moment, right? So they don't have lung nodules. They don't have any indication that a radiologist would say, okay, this person is at increased risk for lung cancer. But we can, of course, quantify the biological age much better than a human can. So is this information useful? The nice thing is that such a system can really run completely automatic at the background, right? Really like an ambient screening kind of setting. So we developed the system using the PLCO data set, and then it only included parameters that are very common in the EHR, right? So we can plug it in. It just takes the X-ray image. It also takes age, sex, and smoking status. And then it outputs a probability for 12 years survival, lung cancer, right? So will this individual within 12 years develop lung cancer or not? And again, these very commonly used parameters that are very easy to acquire in the medical record. So we can really plug it in as an fully automatic screening tool. And then if you then look at the results, you can see that we again could find a very at-risk population for lung cancer. So the nice thing is that these at-risk populations can now be included into a low-dose screening lung cancer screening program, right, where they get annual scans or even 2 scans a year to just rule out that they have lung cancer over time. If indeed we would use such a system to select these high-risk individuals and push them into the lung cancer screening program, we would see that the program would become 30% more efficient in finding more cancer, so finding 30% more cancers while with the same number of scans, right? So with the same cost. So and a lot of these individuals were not eligible for low-dose lung cancer screening at this moment, but were of increased risk that we could then identify. So this is just to give you an idea how such a system, ambient screening, could be implemented in the clinic, and how we can identify individuals of risk for cancer or cardiovascular diseases. So what I showed you before is, complex algorithms that try to quantify aspects about your face age and your chest age and your biological aging. But of course, also what we can use AI for is to automate known predictive factors. So say, for example, we want to use AI to predict cardiovascular risk, right, from a CT scan. So the nice thing is that we already know what is important there. So if you look at the coronary artery classifications, CAC, and these are orange, so in blue is the outline of the heart, and in orange you can see in the coronary arteries. If there is calcium in this, which you can easily see on a CT scan, then this is a very strong risk factor. It's the best known predictor of cardiovascular events. The nice thing is also this guides from primary prevention therapy, so we know what to do, right? If individuals are at increased risk of CVD, cardiovascular disease, well, we know how to act on this and how to what to do with these individuals. It is also visible on any chest CT, non-contrast and enhanced chest CT, but it requires a lot of expertise and specialized equipment and a human to look at this. So to calculate this risk score, this CAC risk score, it is simply too expensive to implement this for every scan that we have that is made in the hospital. So there's a lot of individuals that are now getting scans in the hospital that are in the data. We can very quickly see that they're increased risk for cardiovascular disease and that we should act on this. But this information is completely not extracted simply because it's too expensive in the current workflow. So what we said, okay, let's try to completely automate this whole process. So we localize the heart in the image, then we do a very detailed segmentation of the heart, then we look at the coronary arteries, then we see if there's calcium there, we quantify the amount of calcium, and then we do a coronary artery calcium score, like a risk score. And this is fully end-to-end risk score that automatically runs on our clusters in under 2 seconds. And very similar to the examples before, this really allows for the high-risk identification, for the identification of high-risk individuals in organized and opportunistic screening settings, right? So you can put this into screening settings for patients that were screening for lung cancer with a low-dose CT, but you can also put it into a hospital where any CT that a non-contrast CT that's being made is being analyzed by such a system. So we had the Framingham Heart Study, which is high-quality CT data for training. And we had cardiovascular radiologists providing the ground truth for all of these, and then we used that to train the algorithm. And then we used over 20,000 individuals from various cohorts that are asymptomatic and symptomatic to validate our algorithm. So we had the Framingham Heart Study, but the second screening round, which was performed a couple of years later. We used the NLST, so the National Lung Screening Trial data for validation, as well as 2 symptomatic cohorts of patients with stable and acute chest pain. And I think, so the first step that you do is you look at inter-reader agreements. So how does the AI agree with the expert readers? And then over 5,500 individuals that we had both data types available, we found a very high concordance and a similar concordance between 2 readers, right? So the AI algorithm did it as well as a human did in this case. So for the cancer, because I understand this audience is more interested in cancer. So if we then look at the National Lung Screening Trial data, we could immediately see that we only using this measure, that there was a very strong stratification for overall survival, only using this CAC risk score. Right? So if you have a high risk score, there was a very significant drop in survival for these individuals. And this also, again, remains statistically significant after correction of the clinical data that was available in LST. But what is, I think, very interesting here is that if you look at the lung rats, and these lung rats are the clinically used guidelines for predicting who will have cancer from lung cancer from these low-dose CT scans, you can see actually that the risk of the high-risk CAC risk group is very similar to the high lung rats risk group. So by doing so, you can really see that it's almost as important to look at the heart in lung cancer screening than it is to look at the lungs for lung cancer. So really try to get more data out of these data, more information out of the data that's already being acquired for various medical purposes. We can also, and this is something that we work a lot on, and I won't bore you with all the details, but we also very often look at like, okay, where does it go wrong? Why does it go wrong, right? Is there a pacemaker or not? Is it a stent or not? How stable is this algorithm against these different things? How can we improve the algorithm? Because it's simply, there's too many scans to look at. So it's very difficult for us to check all the outputs of the algorithm. If we, do say 70 or 80,000 scans, which is something that we're now seeing in our hospital annually. So we have to make sure, and we have to check and control that these algorithms work well, but also keep on working well over time. All right, so now I'm going to give you another example how we can use AI in medical imaging and the problems that we encounter there. So this is a clinical problem, right? Lung cancer segmentation. So you have lung cancer, and there's involved nodal disease, and you're being referred to radiation. And more than half of all the cases, lung cancer cases, get radiation. So then we need to define the extent of the lung cancer tumor, right? Because that is where the radiation dose is going to go. And if you then have humans, which you can see on the left, you can see that there's a huge intra and inter-observer variability, right? So 2 doctors will not segment the same volume. But also the same doctor over time will segment a different volume. So the ground truth here is very unstable, right? So what we said is, okay, let's develop an AI contour and let's see if we can better harmonize this. So at least we get to more similar segmentations, but also if these segmentations are closer to a certain individual that has a lot of experience with this and perform very detailed segmentations, much more than in clinical practice, on a training data set. So we developed this AI tumor auto segmentation module, and this was very recently published in the Lancet Digital Health, where we had many different data sets that we combined into this analysis. So first, we had 3 different cohorts from Europe and the US, where we had an expert segment all of these cases and provide a single expert and provide the ground truth for the algorithm. So this algorithm was really trained towards one individual that we thought had the most experience and performed that well. And then we had various data sets for primary validation and over 1,300 patients from data sets from the US, Canada, and China. And the nice thing is then we can do a lot of functional validation, right? We can look at how these segmentations, does it really matter for the clinic? How stable is this algorithm for test, retest, and different seed points, which are often initially, so different starting points for the algorithm. Does this matter for the end result? But also we did a lot of end user testing. And that is, I think, where it became very interesting, is that how physicians were actually working with these different algorithms. So, and I apologize already up front for this figure, but there's a lot of information in here, but I'll try to walk you through it. So what you can see on the left, the purple and the red, these are the benchmarks. So these are the humans against each other, and this is intra and inter observer. So this means the same individual doing a couple of things twice or even more times, which is the purple, and the red is the variation between different individuals. And the y-axis is DICE, this overlap fraction. So how similar are these segmentations? So then, in the brown is an older algorithm that we use that is suboptimal, and then the new algorithm that we developed is the blue, orange, green, and so it's showing purple on my slides, but I'm not sure how it's displayed. But so these last 4 columns are the new algorithm that we developed. So what you can see is in the blue is that this is for primary tumors only, right? So for the primary tumor lesion, we found that actually, if humans were using them, is that we were significantly reduced the variation between them, right? So they got this algorithm, this output as a prior. So it was presented to them. Then they needed to change this to what they liked and then approve it. And we saw that they're actually that there was a lot of harmonization between the human experts in this regard. But what is really interesting is in the green and the purple at the end is that if we look at the nodal disease, right, the lymph nodes that were involved, and also the segmentation that were provided there, is that the algorithm was actually not performing that well, and it was not helping the clinical routine. So, we did detailed investigations about failure mode, right? So where did it fail and where did it go well? And, we saw that the algorithm under-segmented in certain cases and over-segmented in certain cases. So what this really helped us to provide these caution or warning labels that should go with such an algorithm, right? So, model performance of this certain model is not going in this specific clinical scenario. If we would do it like this, then also a user will know, okay, I have to be very careful because there's a collapsed lung, and we know that the algorithm doesn't work that well with a collapsed lung, right? So it trusts the outputs less. So this information, these warning and caution labels are very, very crucial. Then we actually went on and we said, okay, now we have all of these lymph nodes, right? These lymph node stations that are involved. And this is on the x-axis on the bottom. And then we had the train, tuning, and testing data sets. So we said, okay, we know that they don't work that well for certain lymph node stations. Can we actually increase the different data sets for training and give the algorithm more examples of how they look like and how they should be segmented? And do we then also see that the performance went up? And indeed, that is something we saw. It's also logical, right? Like the more examples the algorithm has to learn from, the better it performs at that specific task. So we could really say, okay, now tailored, right? Like we see a problem for this lymph node station, let's increase the training data sets. And we see indeed that in the testing data sets, that then the performance went up. But also for other lymph node stations, we simply did not have the data. And this is also something that is really important, right? Another caution and warning label is that if there's a lymph node affected at a certain location, then be very careful to use this algorithm because you cannot trust this output because there was simply not enough training data available and the performance is lower. So these kind of investigations, which are very time consuming, I think it's very important to really move these algorithms into clinical practice. Next, we did an investigation about end users. So how do radiation oncologists and oncologists like to use this? And what is important there, and for the sake of time, I will go over this briefly, is that the human AI collaborations, if they would work together, if they in this has a very similar performance to humans alone, but it reduced the variation by 32%, and it was 60% faster. So with a similar result, we saw that they could get much quicker there, and also the variation between individuals became much smaller. So this means that patients are treated more similarly, which is something that, of course, we want to have. Also, at the end, we asked them, how do you actually perceive the AI quality? And this is really interesting. So de novo, so before AI, so without AI, we saw that 70% of the cases were found to be very challenging. Challenges are very challenging. But the moment that AI assistance, so the AI assistance was introduced, we found actually that only 29% of the cases were still challenging, and that the rest was neutral or trivial. So this really means that if you use AI, that actually the job of the oncologist becomes easier, right? So I find less challenging, which is also a good thing. And they liked using these tools in clinical practice. And this is also something that we tested in terms of efficiency. Do you find this efficient in terms of time savings? Do you would like to use these tools? And the vast majority of cases and individuals, they agreed that an AI tool was actually useful for a specific use case. So these are examples how we can use AI in certain specific clinical settings. Just to give you an idea, is that we can also go beyond what humans can do. So this is like a field that we call radiomics, where we extracting a large number of characteristics from these tumor lesions, and this goes beyond what humans, more than meets the eye, right? So more than humans can extract from this. And we see that, so we develop three-dimensional CNNs to extract all this information from the tumor region and then provide predictions. And this can be, does this lesion respond to cancer treatments, IO treatment, or chemo, radiochemo, or targeted therapies, but also what is the underlying biological patterns, right? What is the mutational patterns and the mutational load of this lesion? All of these different aspects we're trying to now investigate. And we indeed see that, very in detailed experiments, again, using a large number of different data sets, we see that these models have a very strong performance in various scenarios. So these are lung cancer patients, for example, that are treated with radiotherapy and surgery. And very similarly, we see also very high performance for predicting IO response. So can we predict for a lesion how well they're doing, how well the AI treatment will work. The nice thing is that we have also multiple time data available, time points available for a lot of these cancer patients, so we can integrate them into more complex deep learning networks where we take all of this time information and then improve predictions. And indeed, we also see that adding more and more data to the deep learning algorithm make it actually better than a single time point. So there's really value in combined learning of all of that data and really do this time tracking and monitoring and response prediction for predicting who is actually responding to cancer treatments and who is not. All right, so only a couple more minutes. So what I want to talk about next. So in the beginning, I talked a bit about the why AI in cancer medical imaging and in cancer imaging. Then I gave you some examples how we are applying this. And now in the end, I want to give you some recommendations. So a very big problem in the field is reproducibility. And this is really hampering progress significantly. So this is a paper actually that was published already 4 years ago in Science, where they did an investigation of 400 papers at major conferences, and they demonstrated that not a lot of people are sharing codes. Test data is also not very frequently available, and then only half of them have pseudocode. And pseudocode, and is what they meant with this, is a detailed description in the methods that it can actually, so that other investigators could have a slight idea what the people were doing. So they could not reproduce it, but at least they had the methods were detailed enough that they know which functions were used and such. But this was only available for half. The other half didn't provide sufficient details. And this is something that is still today, and this is also already a year old, but all of the reports are showing this, right? Like a lot of these papers are hit and run. So they publish a paper and they run to the next problem. They try to publish this and they do, and there's not a lot of longevity there. They don't answer questions. They don't share code. They don't share data. So this is a very big transparency and reproducibility problem that we're facing. And this is also and this is a paper that made a lot of impact that was published recently, where they evaluated all of these publications that came out to use imaging data to predict COVID, right? Like, does this individual have COVID or not? And also, can we use this to prognosticate how well this individual will do over time? And there was about 20 to 25 papers, if I'm right, that were published in this field, also in very high impact journals. And the authors concluded that actually none of them will likely make it to the clinic. Also because there was almost no methods described, there was nothing shared, there was no reproducibility or transparency of the authors, and also there was experimental design problems and all of these things. But it's really interesting that they also again found, and that's something that the COVID era actually learned us, is that reproducibility and transparency is really hurting the field. So we wrote actually a response to a specific paper, and that was published by Google, about the need for reproducibility and transparency, and things that are very, very common to the bioinformatics and genomics community, like sharing reproducible pipelines, sharing the underlying code bases, sharing the underlying raw data and derived data. All of these things that are very common in the genomics field are very uncommon in the clinical AI field. And that is something that we're really repeating the same mistakes that we did in the genomics field about 15 years ago. And also, these AI models are very complex. And that is also something that one of the statements we made there is that more complex methods demand greater transparency, right? We need to know more about them simply because there's so many different factors that actually influence a specific model. And that is something that we're now working on a lot. So we said, okay, it's very unlikely that medical AI will progress until we manage to this lag of transparency and reproducibility, but also to provide an easy process for others to share models to the cloud, but also to use then these models that are shared by investigators and to show the generalization of these models in other unseen data. One step in this is that there's a lot of guidelines now out there, right? Like many of them, and every month there's a new one, which is really great because we are getting to these different guidelines that are helping to standardize and what we should share and what we shouldn't share. But we said, okay, we want to take this one step further, and this is a cloud-based platform that we're currently developing. It's called modelhub.ai, where we want to provide a platform, an easy platform for investigators to have an end-to-end sharing of a reproducible AI pipeline to the community. And how it works is that, in a regular workflow, you have all of these specific steps in the design about, defining the research questions and choosing different data sets and model architectures, then do the analysis where, we have to think about hardware and software and all of these are different aspects that need to be set up. And also, we then do an analysis and then we have reporting, right? So document the code and the environment and all of these different things. And what we now do is in the cloud, we use Google Colab. We take advantage of the cancer research data commons platforms and developments, where we really want to have these end-to-end reproducible pipelines, where a lot of things are actually much easier, right? We don't need to set up hardware, we do not need to set up software. All of these things are being taken care of you in Jupyter Notebooks or Google Colab environments, where it's very easy to access these different models and to also upload them and also evaluate them on different data sets that are available. So first, several of these model hub are several of these models and reproducible pipelines have been shared. So you can go there and let me know if you have any questions about them or you would like to use them. And we expect many more models to be shared there over time. All right. So at the end of my talk, and thank you for your attention, and I'm looking forward to the questions.
And thank you, Dr. Aerts, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located at the far left of your screen. So let's get started. It looks like we already have some great questions coming in. Our first audience member says, I have a question about the explainability of these algorithms. Is this needed before clinical introductions?
Yeah, that is a great question. So there's a lot of people investigating the explainability of AI, right? These are very complex, nonlinear models. How do they, how do they work, right? And so my approach is I'm very data and results driven, right? So if an algorithm, a black box, if it works and it keeps on working under different conditions, then we can trust its outputs. And very similar to a Tesla car, and we can debate if they really work well, but it's pretty amazing what they already do, is that you don't exactly know how this algorithm is working in the background. You can see, okay, it detects objects, but it detects a bicycle or detects another car, but it is very, the process to get there is very nonlinear, very complex. So I see this very similar in medicine, right? Where explainability, if we can, yes, this is very important to investigate. But the most important thing is to really make sure, how well do these algorithms perform today? How well will they perform in the future? If there's changes in the input data, how well will the algorithms again change? And this process needs to be monitored. So that is also the big issue with AI and medicine models, is that a model will be introduced into the clinic, but there's a very heavy processing and monitoring process that needs to be incorporated with many of these applications as well.
Thank you so much. And our next question, how do you see the clinicians respond to the potential introduction of AI technologies?
Yeah, so that is also very interesting. So of course, the clinicians that we work with and AIM, right, there may be a subset and bias from, say, the more average clinician. Overall, we see actually not a lot of resistance against these algorithms. There's a lot of algorithms that really make their lives easier, right? So if an oncologist needs to spend less on defining treatment plans and setting up all the, say, the radiation plans for radiation therapy, and spending less time on this, but more time on, communicating with the patients and doing all of this. This is a very attractive thing for them, making the most difficult parts of their lives easier using the implementation of a very specific AI tool. Of course, there was a lot of resistance in the beginning when IBM Watson said that and they took a very high-level approach where they said, we're going to replace a radiologist, which has been found to be very, very difficult. Of course, these remarks didn't really help the trust of the field in these technologies. So what I expect, and what you also now see, is that very specific tools are now entering the clinic that are targeted at very specific touch points, and that they are thinking, and there's a lot of research and higher level concepts, right? Like how can we really use AI as a second reader of scans for diagnosis, for example. But that is something that is very much further from the clinic, I would say. And I also expect more resistance at the moment, because these algorithms will also make mistakes. And we evaluate AI much harsher than we would evaluate as another human being. So there's some resistance, but overall the responses are very positive.
Thank you so much. And I want to thank our audience for these great questions coming in. Our next question is a two-part question. How quickly do these programs analyze new data, and how often do they need to be updated? And does that make the clinical application difficult if the programs change rapidly?
Yeah, that is a very big issue. and there's also the FDA is also struggling with this. So how do we, make sure that they are working today, but there's a slight change, for example, in the acquisition parameters of a scanner? Do they still work? And that is something that needs to be regulated well, but also needs to be controlled well. So what is now happening is that you see, okay, this algorithm, there's like a whole list of things, of conditions that need to be met before it can be used, right? It can only be used on a specific scanner type. It can only be used on a certain acquisition settings, reconstruction parameters, for example. And all of these need to be met. And if they're not met, then we are unsure if the algorithm performs well. That is in the case, for example, where it does a prediction of, say, cancer risk or future cancer risk or future cardiovascular risk. It's a situation where it's very difficult for a human to actually check this, right? Does it make sense? In other scenarios where it's easier for clinicians to actually see where and when the AI model breaks, we think that we see actually that these requirements become more loose. So if, for example, we need to define the tumor target and the tumor volumes, and the AI model starts to present nonsense, a human will always check this and approve this at the end. So there's a much better control mechanism of a human in this process, and therefore this also requires less monitoring and less control of how well the algorithm is working. And there's, of course, then you can just simply stop using the algorithm or give this information back to the developers. But this is a huge problem. Very small changes to the input data can have huge effects on the performance of models, and this is something that needs to be controlled for and needs to be monitored.
Thank you so much. We have one final question coming in. How would you comment on the utilization of AI in automating the process of a single cell sequencing and multiplexing the biomarker expression profiles in TME if heterogeneity has to be addressed?
So we don't do specific research in this area. But what I know from this, right, like with single cell sequencing and also using these scans, is that it is very, very difficult to always. So say you have very large data sets with a very large number of cells and data and images. How do you make what do you quantify this? So say we are quantifying a certain pattern is always very global, right? Like we see this also in cancer now, where we look, for example, at immunotherapy response, right, and PD-L1 expression and all of these things, right? Like, but there's also variations of these, somatic mutations and RNA levels in expression levels in each of the different samples, and that is something that is very difficult for AI. Of course, they are very good in holistic. So just say, okay, here you have a scan, do a certain prediction. But if you then give them a lot of data, they of course need to have also a lot of training data to learn from. And it will always average, like probably a human does, right? If we would look at every cell individually, then we also have to make, say, a prediction of a certain thing that is a single parameter. We probably will approach this in a very similar way. But again, I'm not an expert in this specific field.
And actually, we actually have time for one more question, and another one just came in. How do you see the role of industry versus academics with regards to clinical AI systems?
Yeah, that is actually a very interesting question, but also something that is so overall, I think it's really interesting now to see what is happening, where you have Amazon and Google and Facebook and Microsoft all in this space, in this clinical AI space, and they're publishing papers also in Nature and in the high impact journals. And it's very difficult also for academics to compete with this, right? But I think there's a very distinct there are very distinct roles for each of these to play, right? Like academics, we're really good in just demonstrating, hey, this is a cool idea about phase age and it works. Wow, that's amazing. And then, but we are not, of course, an academic team is very difficult to push this really through into products, right? We're not app developers. We're not, doing integrations with EHRs or other systems, right? That's not what you do in an academic lab. So I think a lot of these technologies will eventually make it into society, but there's very distinct roles, and a lot of the more clinical integration, clinical evaluation, things that are much more time consuming, and a lot of also academics find more boring. But these things need to happen, and very often this happens in industry. in really driving to develop certain products. So I think there's a very different role for both. And of course, they overlap in certain aspects, but it's very interesting to see now how this dynamic is playing out in the clinical AI field.
Dr. Aerts, this has been a pleasure. Thank you for your presentation today. Would you like to provide our audience with any final comments before we go?
Yeah, no, thank you for your attention. Let me know if you have any questions about any of these things, or you would like to know more about the platform, for example, that we're developing. I'm very happy to respond to this.
Well, thank you again, Dr. Aerts, for your time and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until October 4th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Have a great day.